I am Professor Shuman Chakraborty, a lead investigator of this project, along with the research team of Professor Arindam Mondol from the School of Bioscience. When the COVID-19 pandemic hit globally, that was the first time when the RT-PCR test became a household name. The RT-PCR test requires a highly sophisticated lab in which there is an expensive machine, but more than that, it's not really a one-time investment. There has to be a proper infrastructure that is maintained and run by a highly skilled technician. On the other side, people also became aware of a very simple user-friendly test which could be conducted anywhere with very quick turnaround time that was possible by the rapid antigen testing using a simple paper strip to get the result. We wanted to develop a test which could be almost as accurate as an RT-PCR test but could be disseminated in a decentralized manner by frontline health workers or unskilled personnel completely outside controlled lab. Our researchers, including professors and students, were looking into various possibilities of coming up with alternatives of the RT-PCR test. We came across the possibility of using RT lamp as a technology where the advantage could be that instead of the real-time PCR machine which requires thermal cycling, one could use isothermal or constant temperature processes.
We have developed a PCB which can do that particular thing at a very affordable cost. The first version of the PCB was quite crude, after which we did several tests and identified ways to make the PCB further smaller. This was the second generation PCB that we've developed, after which we started working with SMD components, which results in smaller PCBs and finally culminating in the PCB that we've been using in the present device, which is the size of a credit card.
At the end of the reactions, you have to make a detection, and normally in a real-time PCR machine, this detection requires very sophisticated optical system. We tried to improvise upon that requirement to make this entire detection smartphone interfaced and smartphone enabled by performing automated image analysis and image classification and quantifying that in terms of positive or negative infection by using training data sets that we get from RT-PCR benchmark test results. After the sample collection, within 45 minutes, the entire test could be conducted by unskilled operators in a single user step process with no chance of contamination. By the time we developed these technologies from the lab to the engineering prototype, we validated this in different ways. The entire test was validated on around 200 numbers of patient samples, and the early results were extremely promising and were certified to be amenable for further development towards commercialization.
For developing this particular test, we have standardized multiple biochemical reactions rigorously, and then we have integrated all these different biochemical reactions in a single integrated step that can be performed by any unskilled laboratory personnel using the COVIRAP machine. We not only stopped by developing the assay system, but we are also standardizing all these reagents at our in-house facility by preparing the enzymes, the buffers, the probes, everything in our lab. And then we are assembling all these components of the reaction mixture together in the form of a kit in our in-house kit preparation facility before they can be used for the patient sample testing which is going on in our BC Rai Technology Hospital.
We got an authorization from the Institutional Ethical Committee to perform field testing of this particular technology outside the lab in the primary healthcare center, the BC Roy Technology Hospital of IIT Kharagpur. Huge interest was created as this very unique technology, which is a stand-alone technology on its own, where an extremely user-friendly, low-cost device unit implementable in a community setting for nucleic acid testing for the first time being presented in a rapid testing format. There were industrial licensees who transferred this technology from IIT Kharagpur for developing a commercial version of this device and at the same time commercializing the development of the kits. So that has led to the subsequent lap of the race, where the commercialization phase is currently underway. The commercial version of the prototype has been developed. The commercial version of the kit has been standardized. It is being commercialized by companies in India and abroad. So a wide number of tests, for example viral, bacterial, parasitic infection, could be conducted using this technology, which we have given a generic name as PNAT, piecewise isothermal nucleic acid testing technology. Currently, there are no such competing devices of this platform technology nature. The device worth 8,000 to 10,000 rupees is a one-time investment as opposed to the 20-25 lakhs rupees of the real-time PCR machine, and in addition to that, all what you require is a smartphone.
One of the key features of this test is basically the primers and probes which are responsible for detecting the viral RNA. We have designed these probes and primers in a generic way so that they can detect all different variants of the SARS-CoV-2, which is the causative agent of the COVID-19 infection. These primers has been able to successfully detect different variants of the SARS-CoV-2 like the Delta and the Kappa variants in a recurring way during the ongoing field trial which is going on right now in the hospital.
Atmanirbhar Bharat, which has been the dream of our Honorable Prime Minister for long. The Atmanirbhar Bharat or the self-reliant India, this paradigm cannot be achieved just by reverse engineering or copying technologies from others and simply adapting that for our need. It all can be made possible via disruption in the technology development, dissemination, supply chain and integration of that with the grassroots level disease management, so that the underserved community, the under-resourced community can get the maximum benefit. We can eradicate the difference between the haves and have-nots, those who are not able to afford this test and those who can afford high quality, expensive diagnostic tests. We believe that this technology, which is now under commercialization phase and in field testing, it has given results of extremely high accuracy as comparable to the RT-PCR test, and it is ready to get into market not just in India but in several other countries where this kind of technology appears to be a missing link between the high-end, high-cost nucleic acid tests and compromised accuracy yet simple rapid antigen.